kind of nice going later in the talk because I can rewrite everything after I hear what other people say. I want to introduce the research team here. This is a project we just started last year in 2025 with funding from the NAPI Foundation, and we appreciate that. Dominique and Emily are both here in the audience, so feel free to ask them any questions. Zach's running all our cyanobacteria toxin analysis, and Anne's been helping us out in the lab. Just a quick bit of review. Our real question is, why are we getting blooms? And I think that's everybody's questions. And the thing you need to remember about a cyanobacteria bloom is there are 5 or 6 things that are important for the bloom to happen. You have to have enough light. You have to have a warm enough temperature, someplace above 12 to 15 degrees, and you have to have calm winds so that they can control both float to the surface where you can see them, but it's also very important that Microcystis is actually a low light adapted species. So if it floats to the surface, it gets sunburnt. And so it actually needs calm enough winds that it can control itself in the water column in order to not get too much light. Mike talked a little bit about these physical parameters. I'm not going to talk a lot about them. You obviously need a source of food, nitrogen and phosphorus. And then you need seed population, and there's this whole question about zebra mussel grazing. Dominique mentioned a little bit about the zebra mussel grazing. The seed population since 2017 is a non-issue because we've had a massive bloom, so there are seeds in that lake for any bloom that we ever want to have. So I'm going to focus on nutrients. And we really had 2 questions. The first question was, what's happening in the lake, in the algal population before the bloom? I sort of like to use the analogy of a car wreck. If you see a car wreck, the bloom, you're not going to know what led to the wreck, right? You got to look at what happened before the bloom to understand why the bloom occurred. And so we approached this by doing weekly sampling at 3 sites, and I'll talk to this. And then the second question was really more, we assume these blooms are regulated by nutrients and physical conditions. And so if nutrients are important, what nutrient is actually driving this? And I'll talk a little bit about nutrient levels and what nutrients drive the bloom. So let's go to question 1. This is the weekly sampling looking at populations in the lake. And we picked 3 locations. We had the Sailing Club down on the eastern side. We had the Country Club Pier up here, and then what we call the North Shore site, which is basically right outside the Skaneateles Lake office off their breakwall. These were chosen, not necessarily the best sites in the world, but they were chosen because they have a historical report of harmful algal blooms, so we thought we might get a bloom at that. More importantly, we could get to them easily because we had to be able to sample all 3 sites at basically within 30 minutes of each other. So we had to be able to get around to the lake to get a quick snapshot. And we had to get them back to the lab within an hour or an hour and a half for processing because we knew there was a holding time on some of the measurements that we were looking at, right? So we collected weekly samples from June through October through these 3 sites. And this is some of the data. Once we took these weekly samples, we brought them back in the lab and we ran them through an instrument that's called the fluoroprobe. And the fluoroprobe, what it'll do is it'll give you the total chlorophyll in the algae, and then it'll break that chlorophyll down into how much chlorophyll is associated with green algae, how much chlorophyll is associated with diatoms and dinoflagellates, and how much chlorophyll is associated with cyanobacteria. It gives you a few other things too, but that's really what we were interested in. And I'm going to show you a bunch of graphs. They're all set up the same way. On the left side is the average of the 3 sites. And so if you see really huge error bars, what that tells you is the 3 sites were not necessarily all doing the same thing. And then on the far side, you'll see the 3 sites shown individually, right? And so you can see here, starting in June, running through October, the chlorophyll in the water column is pretty low, and Bram talked about this too. Satellite imagery also shows this. The chlorophyll in the water column was pretty low, but it gradually is increasing over the course of the season. If you look at the individual sites, you can see there's more dynamics there. And I want to call your attention to two sites on the North Shore that I've got marked with August 5th and August 12th. And you can look at this site and you can say August 12th is higher than August 5th. So this bloom is going up. But you get a very different interpretation if you start to break it apart. And so this is actually looking at just the cyanobacteria channel in there. And you can see cyanobacteria start showing up around August 5th. They gradually increase up through September. But if we go back on that North Shore site and we focus in on August 5th, you can see on August 5th, we have a huge peak in cyanobacteria. Well, it's not huge, it's 1.5 micrograms per liter, which is pretty darn low. But anyway, we have a peak in cyanobacteria. But by August 12th, even though the total chlorophyll is much higher, cyanobacteria have dropped out of that sample. And that chlorophyll that we're seeing on the 12th is totally due to green algae. There's very little contribution from the cyanobacteria. And it points out some of the difficulties you run into when you use an integrative measure like total chlorophyll to try to understand population dynamics amongst different species, because they actually behave quite differently. You also see we got a very nice bloom at the North Shore on late in September, and I'll talk a little bit about that. Okay. What happens with cyanobacteria toxins? Of course, I'm a toxin lab, so we have to run our cyanobacteria toxins over this. And you can see cyanobacteria toxins. And by cyanobacteria toxins here, I'm talking about microcystins, the liver toxin. We measure the neurotoxins and all sorts of other things. They are all absent. Fortunately for Skinny Atlas Lake, it's purely dominated by microcystis, which produces a single type of toxin, a single class of toxin. But you can see there are peaks and valleys in the toxin production. And you can see that on the left side, you can see the two peaks have huge error bars, and that's purely because the North Shore had a very toxic bloom versus no toxins found off of the Sailing Club or the Country Club. And it again points out some of the problems that you have when looking at different sites. These blooms can be very localized on where they're happening. Just for kicks, we took the cyanobacteria chlorophyll and we took the toxin concentrations, and you can calculate what's called a cellular quota. Normally you would do that on toxin per cell, but you can also do a chlorophyll-based cellular quota, so you get amount of toxin per cyanobacteria chlorophyll. The cellular quota for toxin per cyanobacteria chlorophyll was about 0.12. And very interestingly, throughout the whole mid part of the bloom, it was pretty constant. And so there was very little variation in the toxin per cyanobacteria chlorophyll. We know these blooms are dominated by microcystis, right? We know that in culture, microcystis has a toxin per chlorophyll ratio of about 0.10 to 0.15. And so what this tells us is that the changes in toxicity that we're seeing here are much more a reflection of the changes of biomass in the column. We're seeing more toxin because we have more cyanobacterial biomass, and not more toxin because we're shifting from a less toxic to a more toxic species. All right, so here are my take-home messages. Chlorophyll levels in Skinny Atlas Lake showed a general increase from June through late October. I didn't actually point out, we also have temperature sensors in the water here that are measuring at 15-minute intervals. So during the early part of the season, the average lake temperature increased from 13 degrees, that's really below that 15-degree threshold, up to about 26 degrees in July, and then it sort of stabilized for a while. It sat around about 26. All that earlier chlorophyll growth is dominated by green algae and diatoms. You get around August 5th, the cyanobacteria bloom starts, and it runs up through about October 8th. Very interesting and not what I expected. The temperature in the lake is actually decreasing at that point in time. So it's going from 26 back down to 19. So it's not that we're getting more cyanobacteria because the lake is warmer. It's that we're getting more cyanobacteria because we're above that 12 to 15 degree threshold for a longer period of time. Right? Dominique talked a little bit about this when she talked about that 15-degree threshold. This is a really important concept now when we start talking about bloom dynamics. The blooms are dominated by microcystis. I'm not going to say much about that. Toxin concentrations were variable and low. And we talked a little bit about the toxin quota. Frank very nicely gave us 3 very high toxicity blooms that occurred in Skinny Atlas Lake. We looked at those also. The toxin quota and those blooms, very similar to what we saw. They're all around the 0.13 to maybe as high as 0.5. So we're not seeing a more toxic species. We're just seeing more cyanobacteria in the water column. All right, let's move on to the fun stuff, the nutrient stuff. So we really wanted to know what nutrients are driving the bloom. And so the way we did this is that we would go out in the bloom and we would take 50 some odd bottles and we would fill them with lake water, and we would spike 12 of those bottles with 5 times the ambient concentration of nitrogen. We would spike 12 of those bottles with 5 times the ambient concentration of phosphorus, 12 of them with both nitrogen and phosphorus, and then 12 were left by themselves. And we would put half those bottles out in a cage and we would float it out in the lake, and we would come back after 3 and 7 days and collect half of them. And we'd take half those bottles and we put them in an incubator in the lab under optimal conditions of light, temperature, and no wind, right? And then we'd analyze them again by the fluoroprobe for toxins, and for the phytoplam, which was looking at photosynthetic efficiency. I'm not going to talk about the phytoplam at all, just for time reasons. Okay, so here's a picture of what it actually looks like. This is Dominique sitting out on the dock. You can see the 50 bottles spread out there, which she is diligently adding water to, and then going through and spiking. They're all color-coded with little zip ties around them so we know who is what. They get flipped upside down in this cage. The cage gets sealed with a whole bunch of zip ties. We went through, I think, 300 zip ties over the course of the summer because you have to cut them off and throw them away. They go on the little buoy that's off the top. This is on the Ben Wiles Pier, and they get pulled out, and then they sit there, and we come back. All right. This is the North Shore sampling. Total chlorophyll in blue, cyanobacteria chlorophyll in green. This is the same data that you saw before. The only thing is the red dots I have put on where we did the nutrient experiments. Now, recognize we did the nutrient experiments off the Ben Wiles Pier. This was sampled along the North Shore. They're not exactly the same site, but you can see in most cases they agree pretty well. And the position of the orange dot is actually the total chlorophyll in the control sample when we started the experiment, right? So I'm only going to show you results from 3, a pre-bloom experiment, an early bloom experiment, and a late bloom experiment. So here we are in the pre-bloom experiment. And again, everything's set up the same way. At the top, you'll see the control, the plus nitrogen, plus phosphorus, plus nitrogen and phosphorus using the total chlorophyll. This is all in the lake. The next one over is going to be the green algae. The next one down here is going to be diatoms, and the next one over there is going to be cyanobacteria, and we'll go backwards here. We're in June. There's no cyanobacteria in the water column, so we're not seeing cyanobacteria using the fluoroprobe. That's not saying it's not in the water column. You actually can see it in net tows. It's there, but it's below what we could measure using the fluoroprobe. If you look at the diatoms, you see the diatoms are stimulated by phosphorus, and the addition of nitrogen really doesn't make that much difference. Same thing is true for the green algae. They're stimulated by phosphorus. Nitrogen really doesn't make that much difference. And so we have a classic phosphorus limited situation at this point in time. If you move into the pre-bloom or the early bloom, well, now it gets a little bit more complex, right? If you look at the chlorophyll, you see a certain amount of stimulation from phosphorus, but you see even more stimulation if you add phosphorus and nitrogen. Nitrogen by itself doesn't have effect. So what we have now is we have shifted into a co-limited lake where we are being stimulated both by nitrogen and phosphorus, right? Green algae, very classically co-limited. Diatoms, interestingly enough, still don't really care about nitrogen. They're still phosphorus limited. But if you shift over into the cyanobacteria, you see the cyanobacteria are strongly co-limited, and they grow much better with nitrogen and phosphorus than just with nitrogen or phosphorus by themselves. If we move later into the lake, it becomes interesting again. Green algae are still co-limited. Diatoms have started to move into a co-limited situation. And the cyanobacteria at this point in time, they don't care about nutrient addition anymore, right? So whatever nutrients they need, adding more nutrients doesn't really change their growth pattern. And my original thought was, well, we're missing a nutrient. We have copper or iron or something like that. But in the little tiny corner down there, I showed you the results of the lab experiments where we've growing them in the lab under optimal conditions, right? They grew great in the lab under optimal conditions with nitrogen and phosphorus. So this tells me that we're starting to move into the situation where physical conditions are starting to become more important about what's controlling this bloom, right? We were very interested in the impact. There's some studies that we've done, some molecular data that we have that suggests that cyanobacteria actually have some unusual nitrogen nitrogen utilization pathways. And this whole question about you get more nitrogen, give you more toxin, et cetera. And so we decided we would try to sneak an experiment in between these two data points where we looked at the type of nitrogen that we added. If you look at the green algae, you can see green algae, they grow fine with nitrate, they grow fine with ammonia, and they grow fine with urea. So they can easily use all three forms of nitrate or nitrogen. Diatoms, interesting, didn't like ammonia. So for some reason, they definitely did not grow well in our ammonia addition. And the cyanobacteria, they're equal opportunity consumers, right? They can use just about any nitrogen form that got stuck in there. So if we gave them arginine, we gave them nitrate, we gave them ammonia, or gave them urea, all of those were fine for causing increased growth. All right, so here's our take-home messages from that second part. Diatoms and green algae, early in the season, were limited by phosphorus. Nitrogen addition with and without phosphorus had little effect. Cyanobacteria just aren't there, so we don't have to worry about them. As we start to move into August, Skinny Atlas Lake shifts from a phosphorus-limited lake to a co-limited lake. This is very pronounced for the cyanobacteria. Cyanobacteria were strongly co-limited by nitrogen and phosphorus. Much less so for the green algae, and there are reasons, we can talk about that, and not at all for the diatoms. Diatoms remained phosphorus limited at that point in time. By September, we're still into a co-limited state, but now the cyanobacteria, they aren't responding to added nutrients in September at all, right? So we're not seeing any increase in growth, right? By October, I didn't show you that data, but we've moved back to basically to June, where cyanobacteria are gone at that point in time. Diatoms are phosphorus limited, and the green algae source still remain co-limited, right? I didn't show you the data on toxic quotas, but we did these nutrient spike experiments. The toxin quota throughout these different nitrogen forms ranged from 0.1 to 0.15, which is basically the same toxin quota that we saw throughout the whole thing. So the different forms of nitrogen did not increase more toxicity per cell, and the amount of toxin you got was purely amount due to the biomass that was there in the thing. The one caveat I put down here is we just started this project. We've got a grand total of 1 year of data, and phytoplankton populations are hugely dynamic, so it's really difficult to make definitive conditions on 1 year, especially when you're trying to compare nutrients and physical parameters. So we definitely are going to need to repeat this, and we're going to make some modifications, but we're also open to suggestions. But I do want to point out that one of the key take-homes here is when you start talking about these algal responses, total chlorophyll is sometimes a very imperfect measure because the different classes of algae have very different biochemistries and they respond very differently to different inputs and parameters. And so I'll stop there. I didn't put a summary slide. I didn't put a thank you slide in, but anyway, I'll stop. Sure.
I was very happy with this presentation, Greg, because I tried to connect the dots that I achieved through the remote sensing analysis, and that's nicely overlapped with what we have achieved, but I didn't know the reason. For example, you mentioned that by decreasing the temperature, the total algae is increasing increasing. So there is really no relationship between that. But you explained it nicely that it is because already has above the 15 degree or 12 degree, whatever it is. And then we saw the satellite data shows that the increase of total algae from early in August all the way to early October, then it's dropped. I know that you didn't collect data after October, but starting from mid-October to end of November, satellite shows decreases. So I mentioned that because I think remote sensing can somehow address the caveat you mentioned. The model now have been developed. We can backcast it to all satellite data since 2014, since Sentinel-2 is available, and then generate the map and total chlorophyll for that, and then bring it to you for interpretation and see how this trend works. Sure. So that's good. Thanks.